So the previous video we have learnt about the structure of the muscle, every single part and points of it, layers, its different composition, the membrane that it covers. Now today our focus will be on the muscle cells, the myocytes, and its different proteins that are present inside as the myofibrils. So if you remember by the previous video, the myocyte, they can be they compose of a large amount of this myofibrils, or the one which is nothing but the muscle proteins. Now these muscle proteins are the one responsible for the process of contraction of the muscle. Now each and every of this protein, they are majorly of this two type, the actin filament and the myosin filament. Now this actin and myosin, they work together, and ultimately that will lead to the contraction of the muscle fibers, and somewhat it is mediated by the neurons, as the signal coming from the neuron is responsible for initiating this process of contraction. So the focus for today's video will be completely on this myoproteins, these different proteins of the muscle. Now if you remember that every cells, the muscle cells, they have this large number of myofibrils which are covered by the sarcoplasmic reticulum, and also there are large amount of mitochondria. Now their use is what we are going to discuss today. But before moving on to it, we should know this thing first. There are two types of muscle filaments that are present. One is called the thick filament, and another one is called the thin filament. The thick is majorly the myosin proteins, the entire myosin heavy chains, and the thin filament is composed of the actin, the tropomyosin, and the troponin proteins. Now that way they have two color band patterns, and that two band pattern totally forms of the muscle. Now, due to the overlapping of this thin and thick filament, we find that the skeletal muscles, they show these distinct light and dark bands. And the light and dark bands, it represents the entire of the arrangement of these proteins. Now, one functional unit of the proteins is called a sarcomere, which has the actin and the myosin ends, which are present as shown in the picture in this manner, and they are responsible for the main contraction process. Now, when talking about the thin microfilament or the thin myofilament structure, we have these actin molecules that are present, which are globulin proteins formed of mainly G actin and polymeric F actin proteins, and they are present there, bound together with bound together there, and we'll find that it is surrounded by this tropomyosin, which is a double-stranded fibrous molecule, and along with the tropomyosin we find there are troponins which are discretely arranged in the entire structure. The troponin is a complex of three polypeptides like trimeric proteins, the troponin I, the troponin T, and the troponin C, where troponin I has the ability to bind with the troponin components together, all the components together. Troponin T has the ability to bind with the tropomyosin, and troponin C has the affinity for calcium ion. Right there, this calcium ion is what triggered the entire process. Calcium ions are produced or secreted by the sarcoplasmic reticulum when there is excitation of the particular myocytes by the acetylcholine secreted by the nerve. And once this calcium ion is produced, the troponin, it allows the entire process to initiate. But for before going to that, let's move on and let's learn about the thick filament. Now, the thick filament or the myofilament, the myosin majorly are a dimeric protein, and it majorly contributes 55% of the muscle protein. The dimeric protein has got its head portion and the long chain area. The head is actually the region which has this two portion, one ATP binding site, adenosine triphosphate, and another one is actually able to bind with the tropomyosin. Now, what happens is this actin is actually this entire actin complex. It is governed by this calcium 2 positive ion. Actin, it doesn't contract itself. So what happens is the calcium 2 positive ion secreted by the sarcoplasmic reticulum, it leads to the troponin exposing the entire actin. this actin filament get exposed, and once this actin filament it gets exposed, it has this particular site present in it called the myosin binding site. So those exposed sites will allow the myosin to come and bind with it after this calcium 2 positive binding with the troponin. And when this myosin now take up the ATP or accepts an ATP, it goes and bind with the actin at that particular site. Binding there, the like once it is bound, now it will utilize that ATP and will turn the head portion backward, resulting in contraction of the entire unit. That entire mechanism will be discussed in a separate video. But this is actually the structure of the entire muscle, how they are present there, and what are their major activities. Talking about the muscle protein, depending on the composition of the muscle protein, it can be called a red muscle or a white muscle. Now red muscle, they are majorly the muscle which have thinner fibers and are dark in color due to high composition of mitochondria and high composition of myosin present in it. Now when we talk about the white fibers, they are majorly like thicker in arrangement, a bit pale whitish in color, has got less myoglobin to none, and are majorly responsible for short duration rapid contraction. So they are fast muscle fibers, whereas on the other one, the red muscle is actually have a long duration contraction, so they are actually the slow muscle fibers. Majorly our body's extensor proteins, extensor muscles like the relax, like the one in our back, they are red muscle fibers. Whereas the one which moves faster, very fast, the eyes, the muscle that controls the eyeball movement, is actually a white muscle fiber. That way, in our entire body, we have various kinds of muscle fibers that are discretely separate, like present all over, depending on their function. Some are red, some are white, and even some are mixed varieties that can be found.